Welcome to Brain Ponderings. I'm the host, Mark Matson. This will be the fourth in a five-part lecture series on the neurobiology of aging. In the first three episodes, I covered theories of aging, things like biomarkers of aging, comparative biology of aging, molecular and cellular hallmarks of brain aging, structural and functional alterations that occur during brain aging. And today I'm going to talk about how mechanisms of aging, normal aging, usual aging, increase and accelerate neurodegenerative disorders. And to do this, I'm going to focus on three main age-related neurodegenerative disorders: Alzheimer's disease, Parkinson's disease, and ALS. I'll also mention stroke, which is a major cause of morbidity and mortality, and that's age-related in its incidence and severity. As with all of the lectures in this five-part series, I'm showing slides on my Brain Ponderings YouTube channel. And if you're listening on Spotify or other audio outlets, um, you can close your eyes and visualize what I'm talking about, but it's a lot easier to follow if you go to the YouTube channel and see the slides. This first slide shows the age and sex structure of the United States population in the years 2010, 2030, and 2050. And so the way to look at this is on the left is males, on the right is females, and each of these small bars is the number of individuals at that age. So there's about 2 million male in 2010. There was about 2 million male Americans at age 1, 2, 3, 4, 5, 6, 7, 8, on up into the 20s and 30s, and actually pretty much uniform till you get up to about 50. And then, of course, as people start dying because of various diseases, um, the numbers of those ages decline up to about 100. Not many people live to be 100. Now, one thing you see about this is that there's an increase in the US population at all ages, but there's a particularly large increase uh, in the age ranges of 60 to 80, say, and that's the baby boomers reach this age. Now, these individuals at this age are at very high risk of developing dementia, Parkinson's, or having a stroke. In fact, if you live to be 75 or 80, there's at least a 50% chance that you will suffer from one of these three major causes of morbidity and mortality in the elderly. Now, in addition to the baby boomers contributing to this increase in number of people in this age range, more and more people are living into this age range because uh, they may have heart disease and have bypass surgery. And so whereas they would have otherwise died, say in their 50s or early 60s, they're now living to be 70, even 80, and are then at increased risk for Alzheimer's, Parkinson's, stroke. Similarly, with cancers, there's increasingly early diagnosis and treatments, particularly for colon, breast cancer, and diabetes. Um, it's easily diagnosed and treated at least in terms of controlling blood sugar levels. Okay, so more and more people are getting these age-related brain disorders. Now, most cases of Alzheimer's, Parkinson's disease, amyotrophic lateral sclerosis, there's no known genetic cause, and they're called sporadic. Uh, we do know that aging is the major risk factor, and age-related processes, which I'll talk about, are precipitating the onset of the disease and accelerating it once the people become symptomatic. These different disorders differentially affect neural populations in different regions in the nervous system, although there's some overlap. So in Parkinson's, a brain region that's been focused on a lot, and dysfunction of that brain region is associated with clinical diagnosis. That region is the substantia nigra, where the dopaminergic neurons are, and they control body movements via their axonal projections to the striatum. And essentially, those circuits inhibit unwanted body movement, so you get tremor and other motor symptoms of Parkinson's disease. Later in Parkinson's disease, there will be other brain regions affected, and often people with Parkinson's develop cognitive and psychiatric problems as the disease progresses. Alzheimer's disease, one of the first brain regions affected severely is the hippocampus, which is very important for short-term memory and encoding and recalling memory. So the clinical symptoms that lead to diagnosis are related to damage to neurons in hippocampus and connected circuits. ALS affects principally neurons that control movements, that is, motor neurons that innervate muscle cells. And those neurons, their cell bodies are located in spinal cord. And then there's also upper motor neurons shown in yellow here in the motor cortex. They often degenerate in ALS. Huntington's disease is purely genetic. It's caused by mutations in the huntingtin protein, and early on it affects neurons in the striatum that control body movement. Now, there are a small percentage of cases of Alzheimer's, Parkinson's, and ALS that are caused by gene mutations. About 10%, no more than 10% of all cases. Those usually have an earlier age of onset. That is to say, for example, with Alzheimer's disease, whereas sporadic cases of Alzheimer's, the disease onset is until 70s and 80s typically. With certain gene mutations, which I'll talk about, some of these individuals with those mutations become symptomatic in their 30s. Similar with Parkinson's disease, there's quite a few genes in which mutations can cause early onset Parkinson's disease, and ALS also. There's some gene mutations that cause small percentage, and those mutations provide an opportunity to study the mechanisms of leading to degeneration of various neurons affected, because one can make, and scientists have made, transgenic mouse models in which the mutated human gene is expressed in mice. And in some of those models, the animals develop the cellular and molecular pathology and brain dysfunction or spinal cord dysfunction in the case of ALS that are quite similar, although not identical, to humans with those diseases. There's been a lot of focus in studying neurodegenerative disorders on the abnormal accumulation of specific proteins that self-aggregate within or outside of neurons in the case of Alzheimer's disease, and also frontotemporal dementia. Tau, an intracellular protein, its normal function is to control the cytoskeleton, in particular the microtubules that give neurons their shape, and also importantly, move things around along axons back and forth from the cell body to the axon terminal. And when uh, mutant forms of tau uh, lead to problems with these transport mechanisms, mitochondrial dysfunction, for example, not being able to put healthy mitochondria where they're needed in the neuron, and not being able to remove dysfunctional mitochondria via the process of autophagy or mitophagy. Also in Alzheimer's disease, one diagnostic criteria is the accumulation outside of neurons of amyloid beta peptide in the amyloid plaques, the senile plaques. And this amyloid protein can damage neurons, and there's evidence it's contributing to the disease process. Parkinson's disease, alpha-synuclein accumulates inside of neurons, and it self-aggregates. So you get these aggregates that are not removed by autophagy or other mechanisms, and those aggregates can damage neurons. In ALS, a protein called TDP-43, which is actually interesting RNA binding protein, it accumulates in the cytoplasm of neurons. It's normally in the nucleus, but actually it displaced into the cytoplasm in ALS and also someone at frontotemporal dementia. Right. So how do different age-related changes, things that are happening in all of our brains as we age, even if we're functioning very well, how do those age-related changes influence the disease process in Alzheimer's, Parkinson's, ALS? So this is actually from an article, review article I wrote a long time ago, and with postdoc in the lab. So for example, this one example where early on with aging, you can get mitochondrial dysfunction, maybe a modest deficit in ATP levels, increased free radical damage to various cell constituents, proteins, DNA, RNA, membrane lipids. This then, as actually my lab showed beginning in the 90s, can cause a dysregulation of the ability of the neurons to control calcium, the second messenger that's critical for synaptic function, for learning and memory, for the structural changes in neurons, adaptive changes that occur in response to environmental challenges. This in turn can cause dysfunction of the lysosomes and impaired autophagy. And then, for example, you may get impaired clearance of amyloid beta peptide or tau, and altered APP processing, that is increased production of amyloid beta peptide and plaque formation. So on the left here, you have age-related changes promoting the development of the disease-defining pathologies, amyloid plaques and tau tangles. And then when these pathogenic proteins start aggregating and accumulating, they in turn lead to oxidative stress, dysregulated calcium handling, mitochondrial dysfunction, synapse degeneration, neuron death, and of course, the symptoms of the disease. And with Parkinson's, kind of a similar scenario, with age-related changes leading to the pathology on the one hand, and then the pathology exacerbating, accelerating those age-related changes. I'll just mention with stroke, obviously one of the main problems is in the blood vessels. So just like with coronary artery disease, most cases of stroke involve deposition of cholesterol plaques in the cerebral arteries. And if that occlusion gets too much, there's also inflammation there, and you can get a clot formation, shuts off the blood supply, to the region of the brain supplied by that artery, leading to damage, death of neurons in that area, and associated dysfunction. Strokes almost always occur only on one side of the brain at a time anyway. And because one side of the brain controls the function of the other side of the body, a stroke patient will have unilateral deficits in controlling body movements or other problems. Okay, just a reminder from lecture number 2 in this series. These are the hallmarks of brain aging: cellular and molecular, oxidative damage, mitochondrial dysfunction, impaired autophagy, impaired DNA repair, neural hyperexcitability, impaired production of new neurons from stem cells in some brain regions, inflammation, impaired adaptive stress response signaling. So, for example, by neurotrophic factors and other adaptive stress response signaling pathways, dysregulated calcium homeostasis that's associated with impaired energy metabolism and aberrant neural network activity. And then also cell senescence and possibly telomere damage play roles in normal brain aging. And these all may play some early role in setting the stage for Alzheimer's, Parkinson's, ALS, FTD, stroke. Okay, so here's the general idea. Changes with aging can promote protein aggregation and toxicity. And there's an interactive effect where the age-related changes and these disease-related accumulations of toxic proteins exacerbate each other in kind of a self-amplifying cycle. And then there's also impaired adaptive stress responses. And then these in turn exacerbate these processes. So these cycles, once it starts, leads to damage and dysfunction of neurons. Okay, Alzheimer's disease, you get the amyloid plaques. The yellow is amyloid here, and the black is degenerating neuron associated with the amyloid. This is actually a slide prepared from an Alzheimer's patient that Bill Marksberry, a neurologist at University of Kentucky Center on Aging, prepared and gave to me. This particular slide, he's a well-known neuropathologist, did a lot of important early work on Alzheimer's disease. Now, this amyloid beta peptide that accumulates in the brain in Alzheimer's disease, it arises from an amyloid precursor protein, APP, which is a transmembrane protein with one membrane spanning domain. And there are 3 enzymes that cleave the amyloid precursor protein to liberate different proteolytic fragments of the amyloid precursor protein. Now, the most important physiological cleavage is by alpha secretase. The red here is the amyloid beta peptide sequence. The alpha secretase cleaves in the middle of the amyloid beta peptide sequence. And this cleavage, I'll show you a little bit of data on that, but it has a normal function. It releases this secreted APP from the cells, and these secreted forms of APP play important normal roles in regulating synaptic function, neural excitability, and learning and memory. Okay, this alpha secretase cleavage prevents liberation of the amyloid beta peptide because it cleaves in the middle. Now, the amyloid beta peptide is released from the amyloid precursor protein by two sequential proteolytic cleavages. One by beta secretase, which is kind of on the outside the membrane, right on the membrane surface. And then the other is by gamma secretase, which cleaves within the membrane the C-terminus of the amyloid beta peptide. So that those two cleavages release the amyloid beta peptide, which normally in healthy conditions, it's removed from the brain. Actually, you find it in the blood. And there's mechanisms, lymphatic systems, and so on that remove the amyloid beta peptide, and that prevents its self-aggregation and accumulation, along with other factors. For example, there may be certain enzymes that even degrade the amyloid beta peptide. Okay, so then interestingly, this gamma secretase turns out that mutations in the gene encoding the gamma secretase enzyme, which is also called presenilin, presenilin 1, mutations in this gene encoding this presenilin 1 cause early onset autosomal dominant Alzheimer's disease. So if you have a parent that has presenilin mutation and you inherit that mutation, you will get Alzheimer's disease at an early age. Mutations in amyloid precursor protein are more rare causes of inherited Alzheimer's disease, but have been documented. And we can express mutations in presenilin and the amyloid precursor protein in mice, and those mice will get accumulation of the amyloid beta peptide outside of neurons, and as they age, can develop cognitive impairment, neural network hyperexcitability. I'll mention that or go into a little more detail in that in a minute. Now, from a standpoint of normal function of amyloid precursor protein, there's a couple interesting things. One is that it is moved out from its production in the cell body to presynaptic axon terminals. So there's a lot of amyloid precursor protein in presynaptic terminals, and it's moved along microtubules. Now, activity in neurons will cause alpha secretase cleavage, physiological levels of activity anyway, such as occurred during learning and memory. And that secreted APP will bind to presumptive receptors on the membrane of the postsynaptic neuron and reduce or kind of turn off the signal of the synapse. So it's kind of a local negative regulation to make sure that the synapse is not excessively activated. And this was discovered by Katsutoshi Furukawa when he was a postdoc in my lab, and this nice paper in Nature. This just shows recording, putting an electrode on individual hippocampal neuron. And you can see here the neuron fires, you get these big upward spikes. The neuron is firing, and then Katsutoshi added secreted APP to the neuron. And what you see is the frequency of the firing goes down dramatically, and there's hyperpolarization of the membrane. And he had evidence in this Nature paper that this is due to activation of a certain type of potassium channel whose normal function is to restore the ion gradient after a neuron fires. So anyway, the point here is there's a physiological function of the amyloid precursor protein, and the secreted APP plays an important role in that. Okay, so what about age-related factors and how they may lead to the pathology, amyloid tau pathology, neural dysfunction? Now, I mentioned that there's increased oxidative damage to various cell components during aging. Now, one of those processes involves free radicals produced originally from superoxide generated during ATP production and electron transport chain in the mitochondria. And then that superoxide is converted to hydrogen peroxide in the cell, which in turn, in the presence of low amounts of iron and copper, which there can be, particularly with aging and and age-related changes in the brain, hydroxyl radical is produced from hydrogen peroxide. Hydroxyl radical attacks double bonds in membrane lipids such as linoleic acid and arachidonic acid, these are high levels in neuronal membranes. And when oxygen free radicals attack those double bonds, it sets in motion a uh, self-propagating cascade of that's called lipid peroxidation. And that will actually result in cleavage of these lipids and liberation of a particular product called 4-hydroxynonenal. It's a lipophilic product of lipid peroxidation. And this turns out this 4-hydroxynonenal, H&E, can be toxic to cells, including neurons, if it is not removed from them. Uh, there is a way that cells remove this toxic lipid peroxidation product, H&E, and it involves a antioxidant called glutathione, to which this aldehyde binds, and that prevents the aldehyde from binding to other proteins in cells. But the H&E can bind to proteins on three particular amino acids, cysteine, lysine, and histidine. And in many cases, binding, and this is covalent modification, it's not reversible, H&E binding can impair the function of proteins. These are some of the proteins that we and others found are modified by H&E and their function is impaired. Bob Mark and Jeff Keller showed that the sodium and calcium pumps that pump sodium and calcium back out after a neuron fires, they're impaired by H&E. Certain ion channels, their function is affected. Glucose transporter, lipid peroxidation via H&E can impair glucose transport into neurons, glutamate transport, and we think this is playing a role in excitotoxicity in Alzheimer's disease, and I'll come back to that in a minute. Okay, this is evidence from Virginia Lee's lab, and we'd actually shown with Bill Marksberry that the amount of H&E modification of proteins is greatly increased in brain tissue and cerebrospinal fluid of Alzheimer's patients and patients with mild cognitive impairment compared to age-matched neurologically normal subjects. And actually, that was in the 1990s, mid-1990s. And then Virginia Lee's lab showed a similar thing in a mouse model of Alzheimer's disease where the lipid peroxidation proceeds and we think contributes to the amyloid deposition and perhaps the toxicity of the amyloid. We showed, interestingly, there is a genetic risk factor for Alzheimer's disease, and that is the apolipoprotein E4 isoform. So we inherit from our parents one copy of ApoE from one parent and one from another. If we inherit ApoE3 from each parent, or ApoE3 from one, ApoE2 from the other, we're at relatively low risk of Alzheimer's disease. If we inherit an ApoE4 from one parent, we're at increased risk for Alzheimer's disease. And we're doing all this work with lipid peroxidation and H&E, and I knew that the difference between these three isoforms of ApoE, E2, E3, and E4, is in amino acids that are either cysteines or a different amino acid. Remember, H&E binds to a cysteine. So we found that ApoE2 has two cysteines, one in each of those amino acid locations. E3 has a cysteine in one location, another amino acid in the other, and ApoE4 does not have a cysteine in either of those amino acid positions. So we tested the hypothesis that ApoE acts similarly to glutathione in detoxifying H&E, and that ApoE2 and E3 will sop up, if you will, more of this toxic H&E than ApoE4, and that's what we showed in this paper we published in 2000. Okay, now, as amyloid beta peptide aggregates, and particularly on the surface of membranes of neurons, it turns out this peptide, when it's aggregating, has a predilection for certain regions of the outer membrane of neurons that are rich in arachidonic acid and linoleic acid. When that occurs, this peptide, Alan Butterfield in my lab showed this in the beginning of actually early 1990s. This peptide, when it's aggregating on the membranes, causes lipid peroxidation, H&E production, and impairs the function of ion-motive ATPases, glucose transporters, glutamate transporters. And this, as I showed in actually even before we did those studies, renders neurons vulnerable to excitotoxicity. Now, excitotoxicity, as its name implies, is damage to neurons caused by overexcitation. Most of the neurons in the brain, 90% approximately, are excitatory. They use the neurotransmitter glutamate, the excitatory amino acid glutamate. About 8% of the neurons use the inhibitory neurotransmitter GABA as their neurotransmitter. And their function is to control the excitability of the glutamatergic neurons. So in epilepsy, and also in Alzheimer's and Parkinson's, there's an excitatory imbalance where the GABAergic neurons are not able to constrain the glutamatergic neuron activity within normal limits. And I've talked about this in detail, tremendous detail, in my book, Sculptor and Destroyer. Okay, so one of the early findings we had when we started to look at how amyloid beta peptide causes dysfunction and degeneration of neurons, and this is this paper in 1992. This is a time when I was still doing experiments in the lab myself before I had a really big lab. And I found that if I expose cultured human cortical neurons, cerebral cortical neurons, to aggregating amyloid beta peptide in amounts that by itself, that amyloid in those amounts did not damage the neurons. Nevertheless, it made them vulnerable to being damaged and killed by glutamate. It increased the vulnerability of neurons to excitotoxicity. And that actually is a very important finding because subsequent work and ongoing work actually highlights a very important role for excitotoxicity in Alzheimer's disease. So some of that evidence is Alzheimer's patients are 30 times more likely to develop epileptic seizures than are age-matched people without Alzheimer's. Um, Also, we and others have shown that certain excitotoxins that excessively activate glutamate receptors can cause selective degeneration of neurons, the same neurons in the hippocampus that are selectively vulnerable in Alzheimer's disease. And these excitotoxins also cause short-term memory impairment. Interventions that prevent neural hyperexcitability ameliorate cognitive deficits and neural damage in mouse models of Alzheimer's disease and possibly in Alzheimer's patients. In fact, the only drug that's prescribed for Alzheimer's now that slows down the progression of the disease is a drug that blocks a particular type of glutamate receptor called the NMDA receptor, and that drug is memantine. Also, we and in animal models and Steve Cunnane in humans have evidence that this ketone that's generated during fasting, beta-hydroxybutyrate, can counteract Alzheimer's disease process and preserve memory. Yeah, and these are some of the evidence here. And then also recently, Dimitrios Kapagiannis at the National Institute on Aging showed that intermittent fasting has beneficial effects on cognition in older humans at risk for Alzheimer's because they have obesity and insulin resistance. Okay, so I talked about oxidative stress, uh, excitotoxic imbalance, disruption of calcium regulation, and impaired energy metabolism, glucose uptake. Autophagy is also, impaired autophagy is also strongly implicated in Alzheimer's disease. In Alzheimer's disease, there's can be a dramatic accumulation of junk that's not degraded in the lysosomes as it normally would be in Alzheimer's disease. And this shows electron microscope of autophagic vacuoles that normally should be moving to and fusing with the lysosomes and getting rid of their contents. So this junk, some of which is toxic, accumulates in the neurons in Alzheimer's. In this study of Andrew Fang at the NIA, he showed that two chemicals, urolithin A, these are actually natural products, urolithin A and actinonin, which other labs had shown stimulate autophagy, in particular mitophagy, the removal of dysfunctional mitochondria. In this study, the authors showed that these stimulators of mitophagy ameliorate cognitive deficits in a mouse model of Alzheimer's disease. I'm not going to go into detail here. This shows that these interventions are in fact stimulating mitophagy, and that in these Alzheimer's mice ameliorates the learning and memory deficits. This is the water maze test, and further evidence in there shows reduction in the accumulation of amyloid. Okay, so that's quickly autophagy, just one example of going from pathology in patients to animal models to establish some cause-effect role or at least accelerating role of that particular age-related process. Another one is DNA repair, which is impaired in neurons in Alzheimer's disease. That's actually reduced also with normal aging. And if you, in these mice, which have about they have impaired ability to repair DNA. These are Alzheimer's mice, 3X TGAD mice that develop amyloid plaques and tau tangles and cognitive impairment. And when one compromises their ability to repair damaged DNA through genetic means, that exacerbates the cognitive deficits and pathology and synaptic function. So that's in this paper cited here in 2015. All right, so before I move on to Parkinson's disease, I just wanted to kind of put this, at least in part, together where you have age-related problems with mitochondria, inflammation, impaired DNA repair, oxidative stress, hyperexcitability, etc., and interacting with the abnormal protein aggregation process and genetic factors, whether they be risk factors such as ApoE genotype or disease-causing mutations. And so once you have these processes in motion, that's a big problem, and we actually don't know how to reverse it yet. And I think reversing, that is going back to normal, is unlikely. I can't see it actually, maybe ever we'll see. So risk reduction and prevention, early intervention is the approach that has to be taken. And the good news, which I'll talk about in the fifth lecture in this series, is that there are ways to counteract these aging processes and the disease processes. Now, I just did want to mention that there's evidence just in the last 10 years that with aging and much more so in Alzheimer's disease, there are problems in the brain's ability to remove junk, get it out of the brain into the cerebrospinal fluid and blood, or get it to a place where immune cells can, like macrophages, can remove it. So there's this glymphatic system that's shown to be very active during sleep. So the evidence and idea here is that when you're sleeping, which is very important for brain function and health as you age, this mechanism, these toxic substances, amyloid beta peptide, perhaps alpha-synuclein tau, are removed, as well as other proteins. And in Alzheimer's disease, there's an impaired lymphatic system. Another alteration that's interesting in normal aging in Alzheimer's disease is accumulation of long-chain ceramides, which are lipids that are generated from sphingomyelin via enzymes called sphingomyelinases. And in this study, Roy Cutler showed that during normal brain aging, and much more so in mild cognitive impairment and Alzheimer's disease, there's accumulation of these long-chain ceramides in the brain. And then in cultured cells, if we add these long-chain ceramides, it can exacerbate damage and models that are relevant to Alzheimer's disease. And then others have shown, and here's some of the data here, that the bottom line with this is there's a correlation between levels of brain long-chain ceramides and cognitive decline. So the more long-chain ceramides, it's 24 carbon ceramide here, for example, the faster the decline in cognitive impairment. And that's this study. So this is in a longitudinal study. And then if one inhibits this sphingomyelinase that generates the ceramide, that can protect neurons. So that's ongoing work there. Parkinson's disease, again, mostly sporadic cases, no known cause. Some cases are inherited. A lot of interesting genetic work and then cell culture and animal models based on putting the mutant genes. These are some of the that's kind of the thinking on that time frame of progression of the disease. There's a lot of problems going on before the person is symptomatic and diagnosed. And those include oxidative stress, accumulation of alpha-synuclein in certain neurons, and so on. And there's also non-motor symptoms that occur early, and I'll mention that before I go to ALS. And that has to do with the gastrointestinal system, and many Parkinson's patients have a clinical history of problems with constipation. Okay. Also, impaired smell, sense of smell is common in Parkinson's, even fairly early on, and impaired autonomic function. Okay. Now, just here's some examples of alpha-synuclein accumulation in neurons, in dopaminergic neurons in the brains of Parkinson's patients. Now, there is in Parkinson's, in my opinion, pretty strong evidence that exposures to environmental toxins contribute to many cases of Parkinson's disease. One dramatic case of evidence for this was generated by Bill Langston, and who was a head of neurology at a clinic in California, and his residents and fellows, who over a period of just a few days had several patients come to the hospital with symptoms indistinguishable from Parkinson's disease. These individuals were young, in their 30s. They seemed fine a few days before they came to the clinic, and all of a sudden they had Parkinson's disease, essentially, at least from a clinical perspective. And then, so the obvious question is, what do these different individuals have in common? It turns out there were several other hospitals in there that had similar patients with similar time frame, all during the first week to 10 days. What they had in common is they were all heroin users, and they all had recently bought and taken a new batch of heroin. That particular batch of heroin had a contaminant in it. Whoever synthesized it did it wrong, and it had this MPTP molecule in it. And that's what caused the degeneration of their dopaminergic neurons. So additional work with MPTP in exposing cultured neurons, mice, rats, monkeys. MPTP causes degeneration of dopamine neurons and associated motor impairment. And the way this happens is, it's very interesting. MPTP is taken up selectively into dopaminergic neurons through the dopamine transporter. And then this monoamine oxidase B enzyme acts on MPTP, converting it to MPP plus, which then gets into the mitochondria and inhibits complex 1 in the electron transport chain, leading to two effects. One is reduced ATP levels in the neurons, reduced energy levels in the neurons, and increased production of oxygen-free radicals. So this is why the neurons die. Other chemicals in the environment do the same thing as MPTP and MPP plus, that is, inhibit mitochondrial electron transport chain complex 1. Two pesticides rotenone, and paraquat, which have been widely used in agriculture. Individuals, for example, the wonderful immigrants who come in and harvest crops and do other things with crops, spray the pesticides in some cases. Um, they have much higher incidence of Parkinson's disease than do other people who are not exposed to these pesticides. Um, there's evidence for this chlorpyrifos, another pesticide, as being a cause of Parkinson's disease in some cases. Okay, so you have environmental factors, which seem to play an important role, and aging, which plays a major role in development of Parkinson's disease, and these interactions between environmental factors and the aging process can lead to Parkinson's. At the cellular level, not so much different than what's seen in Alzheimer's in terms of what the neuron experiences in terms of oxidative, metabolic, autophagic stress, dysregulated calcium, etc. There's a huge number of interesting findings in molecular geneticists and Parkinson's disease. Mutations in many genes have been shown to cause Parkinson's disease. Alpha-synuclein, Parkin, UCHL1, PINK1, DJ1, LRK1, this kinase. There are families where these mutations cause Parkinson's disease. And what's very interesting about these mutations is they all seem to be the normal function of those proteins that are mutated revolve around two main processes: mitochondrial function and health, and autophagy and mitophagy. So this is very strong evidence that these age-related decline in mitochondrial function, autophagy, mitophagy are contributing to probably the early onset of the disease process in these individuals with these mutations. Huge amount of work looking at the protein structure of synuclein, LRK2, DJ1, and these other proteins genetically linked to Parkinson's disease. I'm not going to go into the details here. If you're interested in cell biology, mitochondrial biology, autophagy, the structure of these proteins and how they interact with either mitochondrial membrane or different proteins involved in the autophagy process. Very interesting work. Okay, this shows with mitochondria. DJ1 associates with mitochondria, plays an important role in protecting against stress. PINK1 plays an important role also in protecting against oxidative stress and in mitochondrial fission, the division of the mitochondria. Parkin, it's very important in mitochondrial dynamics, biogenesis, fission, and probably also targeting dysfunctional mitochondria for autophagy. Okay, one development in Parkinson's field that began quite a while ago now was evidence that the disease process begins in the gut, not in the brain. And that evidence came from Heiko Brack, who he simply used a alpha-synuclein antibody to document the locations of neurons that accumulate alpha-synuclein in individuals that died in different stages of the disease process. And what he found was that in patients in the early phase of Parkinson's disease, the pathology in neurons was much more severe in the neurons innervating the gut in the so-called enteric nervous system than it was in the dopaminergic neurons. And moreover, the pathology seemed to spread, and this has been shown pretty conclusively since in animal models and also some human evidence. Pathology spreads, that is, the alpha-synuclein aggregations in neurons, spreads from the gut to the brain via the vagus nerve to the brainstem, and thence on up to the midbrain dopaminergic neurons and elsewhere. Okay, so this just shows the intestinal wall here, where when you get a colonoscopy, this is what they're looking at to look at polyps, and there's neurons that innervate the gut and control motility, a movement of food through the gut. So in Parkinson's patients, they have constipation because the signals from the vagus nerve to cause movement of the digested food through the gut is impaired. So these vagal nerves develop alpha-synuclein pathology. In humans, there's evidence, very interesting evidence, in some cases of people with severe intestinal ulcers, bleeding ulcers. One treatment was to cut the vagus nerve right by where it innervates the gut, and that's effective in treating the ulcer. But then many patients who had had this intervention for their ulcer, they were followed over periods of many decades, and it was shown that compared to individuals who did not have the vagotomy, those with the vagotomy had reduced incidence of Parkinson's disease. Also, one kind of actually, if you just put alpha-synuclein, aggregating alpha-synuclein in the gut of mice, it can lead to pathology in the brain. And if you cut the vagus nerve in some mouse models of Alzheimer's, Parkinson's disease, that will prevent the brain pathology. Now, ALS and frontotemporal dementia involve accumulation of TDP-43 within the neurons. This just shows immunostaining with TDP-43 antibody in frontotemporal dementia patient cortex and spinal cord. And you see similar thing in the motor neurons in the spinal cord. Now, in the motor cortex of the brain, the upper motor neurons send their axons down to the spinal cord and synapse on lower motor neurons, whose axons in turn innervate muscle cells and when they're stimulated cause muscle contraction. The neurotransmitter used at the synapse here between the upper and lower motor neurons is glutamate, and upper motor neurons receive glutamatergic input from various sensory brain regions that enable you to control body movements and perform actions. There's quite a bit of evidence that excitotoxicity plays a role in ALS. Some of that is, for example, from Jeff Rothstein at Johns Hopkins, who a long time ago showed that in ALS there's impaired glutamate transport, and also one of the drug that's used to treat ALS and slows progression a little bit anyway, enhances glutamate removal from the synapses. Okay, TDP-43's interesting. It's a normally in the nucleus, and it's involved in controlling RNA processing and export of RNA. So remember, you have genes, the DNA, the genes are in the nucleus, and they're transcribed into RNA, uh, and transcription factors stimulate transcription. So then you have mRNA in the nucleus, and it's transported out through pores in the nuclear membrane into the cytoplasm where proteins are synthesized using the mRNA template uh, from the particular gene. In ALS, whereas TDP-43 is normally mainly in the nucleus, in ALS, it accumulates in the cytoplasm outside the nucleus and aggregates. And that's shown here. And these TDP-43 aggregates can promote hyperexcitability, and one can counteract the ALS process in animal models with drugs that block glutamate receptors. Now, I'm almost done. I just want to conclude with one point here that I really haven't touched on, is that I've talked about how pathology in Parkinson's, I guess, is what I focused on, but it's also true in Alzheimer's, ALS, FTD. The pathology, the accumulation of the misfolded aggregated proteins inside of neurons, is propagated transneuronally from one neuron to the next in a circuit. So the pathology and dysfunction begins in certain population of neurons, say in the gut in Parkinson's, maybe in the anterotal cortex or hippocampus in Alzheimer's, then it's propagated to the subsequent glutamatergic neurons in the circuit. Now, one way that this transfer between neurons is thought to occur is via these extracellular vesicles or exosomes. And there's a lot of work showing that one can isolate these vesicles from blood and show, for example, that in Alzheimer's disease, they have a lot of abnormal aggregation-prone tau or amyloid beta peptide, or in ALS, TDP-43 in the exosomes, synuclein in the exosomes, and PD. So these have actually been used, they're being used for biomarkers now for what's going on in the brain. One can isolate vesicles that are coming from the brain into the blood and then interrogate them to quantify the amounts of disease-associated proteins. So you can have a blood biomarker of these disease processes, which may aid in early diagnosis, and clinical trials then in the future will be using these biomarkers to try to intervene early, even before the patient's diagnosed formally by the clinical testing. Okay, then in the next, the last in this 5-part series of lectures, I will talk about environmental factors that counteract aging and may protect against neurodegenerative disorders. Physical exercise, caloric restriction, intermittent fasting, engaging in intellectual challenges, and having these challenges with intervening recovery periods, sleep, eating, rest. And the cycling between challenge of exercise, fasting, intellectual engagement, like we're doing now, and then recovery can optimize brain health and counteract aging. Thanks.